Chin, how you doing, man?
Yeah, I'm good. Sorry about that. My computer just keeps, like a BSOD, what that is? Blue screen,
BSOD, not quite sure what is that.
blue screen of death.
Oh, okay, yeah,
Yeah,
I know what you
it just kept doing that. I had to restart like 12 times.
mean. Geez,
So okay, yeah. How's it
sounds
going?
good. Yeah, glad we're here. I was just asking, when are we gonna be here till 3:30?
When let's see. Yeah, we can do that.
Okay.
Yep.
I mean, I don't know if we're gonna need to be that long, but we'll see. Yeah, I told you I said I was gonna start on the section bank, but some things came up yesterday and some work stuff, so I wasn't able to get to it.
That's fine.
Um, but yeah, I was actually although my plan is before we start, I wanted to just say, like, I really don't want to take because the plan was to go through the BB and then take the full length tomorrow. But I genuinely don't want to take the full length tomorrow because, like, I want to take the full length according to the plan that you've told. Like we should try to at least finish the section bank.
Yeah,
So I'm thinking like we can do that like next week. Like we meet again next week and then try to go over the section bank stuff and then we take the test on Saturday.
yep. We could do that.
Okay. All right, so yeah, this is just the content diagnostic stuff. Yeah, so I was having a little bit of tough time with this. I kind of understood it when I read it again, but I don't know. Something about it just seems a little still unclear.
Okay. So this is just really Le Chatelier. Um, Meaning, so okay, they're saying that this reaction has a positive change in Gibbs, which means it's not spontaneous, right?
Yes.
So it shouldn't proceed uh forward, right? But
Yeah.
it does. And why is that? Well, so okay, first of all, A, if this was true, right, you would have well, they're giving us the enzyme and it's an isomerase, right?
Yeah. So that doesn't work. That's not ATP. So
Yeah, yeah. ATP hydrolysis, like being coupled to that would be like, some
that'd
type.
be kinase, pyruvate kinase, PFK1, and then hexokinase, right? That's the only three.
Uh, it'll
And
it'll be like it'll be a hydrolase. So uh
hydrolysis. Right.
now B, whether it happens slowly or quickly, is a mat is doesn't that's like the thermodynamic control versus like a kinetic control.
Yeah, kinetic control is when the least like the less favorable product is made faster, I think.
Yeah, so thermodynamics deals with stability of molecules. Uh, Kinetics deals with rate, and
Right. Okay.
so this is saying rate. Now, what you chose here, this is also something that happens with rate because, if you add a catalyst, right? Like if you had your reaction diagram like this, reactant product, right? This is your activation energy, right?
Yes.
And you can add a catalyst to lower it, but that's not changing like the energy level of the reactant or the energy level of the product or the delta H of the reaction. It, so activation energy is going to make it just go faster. So D is wrong for the same reason B is wrong. These are kinetic things.
Okay. I see what you're saying.
Now C, this is Le Chatelier's principle, right? Do you remember what that was?
Yeah, so essentially if we have a excess amount of reactants,
Mhm.
if the if the reactions and products are like in equilibrium, it'll shift towards the other side to create equilibrium.
Yes, exactly. So if it's DHAP to glyceraldehyde 3 phosphate, and you just have an excess of DHAP, it will drive that reaction forward.
Mm, okay, that makes sense.
Yep. Yep.
Okay. Gotcha. Is there anything really beside like because apparently this is bioenergetics. I've never heard of this like topic, but I kind of see where like what this term is. But do you think there's anything else besides that? I guess knowing the delta G is like positive, and it is non-spontaneous, negative, it's like non well spontaneous or
So,
positive is non-spontaneous, the other one's spontaneous, all that. Anything else
yes.
besides that or
Yeah. So I gave you the answer in terms of like conceptual. But you can see that in the explanation here that they're using the this I guess version of Gibbs free energy, but the only important thing here is just that when we use Q, because K is product over reactants, but and
hmm
usually used for like equilibrium, but Q is the same thing. It's just when it's not at equilibrium. So as you said,
Right.
it'll whatever stress placed on a reaction will have that reaction reduce that stress to come back to equilibrium. Yep.
Okay. Let me see which one was next. Just gonna keep going. Um,
You hear the news about Bitcoin?
no, I'd heard I think it was going to what 100K or something.
Yeah, it's like it's probably gonna do it today. It's
That's
like
crazy.
so freaking close. I can't believe it.
You got a lot of Bitcoin lying on chain?
Um, in ways, yeah,
Oh wow. Okay, nice.
like that, and like there's also like tons of companies that bought Bitcoin, so they could be ways to diversify that as well. But
Mhm.
pretty exciting,
Interesting.
pretty exciting past month.
Yeah. Um,
Okay, this is
actually this,
the kind of thing that you just have to know, I guess. So like,
right? Okay.
yeah,
Figured that. Okay, I'll just keep going. I made a donkey card and I was like, you know what, I'm
yeah,
just gonna
exactly. That's what I was gonna say.
do
Yeah.
whatever. Uh, this wasn't too bad. Yeah, I learned this yesterday, so this makes sense. Um, oh wait. Is that it? Oh no. Okay, never mind. Okay, yeah, so like I sometimes saw these terms in some of the full lengths and all that stuff, like exonuclease. And I don't know what that like I don't really know what that like what do I need to know about exonuclease activity?
Well, just go at it the way you would go at anything, right? Which is so what I mean by that is you look at the word and you figure out what it means from the word. So if it's a nuclease,
Okay.
it would break up nucleic acids, right?
Yeah.
What if you had a protease?
Yeah, breaks down proteins.
What about this?
True. Brings up lipase.
Yep. And then
I
there's
mean lipids, sorry,
yeah.
lipids.
And then there's also glucosidase,
Yeah, glucose, right, I guess.
which will
Glucose.
break down, yeah, which will break down like a sugar. So
Okay.
nuclease breaks down some type of nucleic acid. What about exo? What does that mean?
I like the outside, but like outside nucleic acids of
Yeah.
yeah.
So let's say you have an mRNA here, right? Um, well, that 5 cap and the poly A tail protect it from exonucleases, which would attack at the ends, whereas an endonuclease would attack somewhere in the middle.
Okay. Is it just like this is getting me like I guess this is me being very specific. Like it's exonuclease literally like the last couple nucleotides or anywhere near? Or is it like is there a
Just think
limitation?
about
You say that?
think about like so we have a strip of let's say RNA.
Okay.
This is inside, so that's where an endonuclease would do its damage or whatever. But these are on the outside, right?
hmm
Think of it that way. And that's where the exonucleases would do its damage,
Okay.
right? So
Then
yeah,
okay. Yeah, if that if that's the case, if it is removing, yeah, I don't know why. A is wrong then, isn't it? Exciting a mismatched nucleotide.
All right, let's see. What's the function of the exonuclease activity of DNA polymerase? Got it. So I told you what an exonuclease is in general, but
Mm-hmm.
uh, in general, meaning that things that are just exonucleases, but DNA polymerase has like its job is to, add nucleotides to like a growing strand. So
Mm-hmm.
its main job isn't to be an exonuclease, but it could act as an exonuclease, and it does that because when it proofreads, um,
Right.
right?
Yes.
So yeah. So let's see. Um, DNA polymerase and its exon repairs DNA mutation. Yeah. So it does it also. So okay. So remember how I said before about how subtleties in wording
Yeah. Yeah.
and also like I always want to give you like multiple ways to get the answer.
Yeah.
Okay. So let's say that like okay, it's a DNA polymerase, which means that it's like involved in what?
In adding nucleotides, taking out nucleotides that are not there, exchanging the RNA
What
from the
what
primers and stuff,
what process?
DNA replication.
Okay. Now, between A and B, there's a word that is different between the two that
Hmm.
let me,
You mean the following and the
yeah,
during?
yeah, yep, yep.
Okay.
So
So,
if you didn't know any of this, right? You know, okay, DNA polymerase. It works to add stuff during replication, right? So
yeah.
it shouldn't be doing things after
Yeah, that would make
replication,
sense.
right?
Yeah, probably during. Okay.
So there's the way where you just have to know it, right?
Mm-hmm.
Uh, and then there's a way where we look at, the subtleties and the way things are worded to figure out the answer. Yeah.
Okay. Sounds good. And these are just a bunch of more definition stuff.
Hmm. That
Um,
seems like you really know this stuff.
yeah, this honestly, I was wondering maybe we can go over this like the progesterone and the estrogen real quick.
Hmm.
And then in the HCG, I recall HCG from a long time ago, but I was like, oh, interesting. I didn't really remember. I was like, LH.
Yeah. So let me get you this.
Hmm?
the so um let me see the question again.
Yeah, yeah, sure.
Maintains the progesterone production of corpus luteum during early pregnancy. Um, so I find here, I'm just gonna look at my notes. Because the thing is that after the the follicular phase grows that follicle, which then can
Right, because like
become
FSH makes the follicle grow, and then the follicle bursts. No, no, the follicle bursts through due to LH, right? I think. No, it's I think so. Um,
uh, okay. So first 3 months, um, the the first 3 months of pregnancy involves that corpus luteum. So yeah, the follicular phase develops the follicle, and that's when you have your, you know, follicle stimulating hormone, your and that makes that follicle, and then ovulation happens, and if there's no pregnancy, then the corpus luteum just kind of I mean, it continues to grow, but eventually it leaves during the menstrual phase. But if there is fertilization, then what happens is that the corpus luteum is made, and the corpus luteum is supposed to continue to make let's see, estrogen and progesterone, and that is a negative feedback loop to the anterior pituitary gland, which was what was, releasing the gonadotropins.
okay,
And
so you're saying that as the follicle releases the egg,
Mhm,
um, the corpus callosum, if there is fertilization, the corpus luteum
mhm.
starts to secrete. estrogen and progesterone to
Mhm.
tell the anterior pituitary to stop.
Yeah, so the anterior pituitary stops after that point. So
Okay.
you have then that corpus luteum that is the thing that produces the um, progesterone.
Interesting. Okay, corpus luteum. Okay, so corpus luteum, interesting. So it can create. Okay, I see. And again, if it doesn't, if there is no fertilization, like right at this phase, like right when it like around here or something, does it just get rid of it at that point? There's no progesterone, there's no, it just like repeats again, or
You're saying if it doesn't get fertilized?
yeah, where does it
Well,
stop?
yeah, so that's when you have the corpus luteum degenerate.
Okay,
Um, but yeah, I was trying to find like a good picture of like a feedback kind of loop thing, but like that's also one of
difference?
the reasons why why what is it? Um why like a pregnancy test, generally speaking, tests for HCG
Oh,
because HCG is going to be the thing that because initially you have your anterior pituitary giving you you know what here let's see kind of
goddamn.
trying to find okay I mean whatever I'll just tell you then um so yeah basically what you have is your hypothalamus releasing GnRH to the anterior pituitary, and the anterior pituitary releases LH and FSH, and
Hmm.
LH um, can cause, the like, well, LH and FSH, like the FSH will develop that follicle for that follicular phase.
Yeah.
And then during that luteal phase, you'll have, if there is, um, if there is no fertilization, it'll just degenerate and leave during menstruation. But if there is pregnancy, then that follicle turns into its own gland, like endocrine gland, that releases
Oh, interesting.
that produces estrogen and progesterone, and that sends a negative feedback to the anterior pituitary and to the hypothalamus for them to stop releasing, LH and FSH. Um, and those things, LH and FSH, like, they can cause that estrogen and progesterone to be released in the ovaries.
Okay.
Yeah. And yeah, and so when you have your corpus luteum, it sends them that, yeah, it tells the brain essentially, or the pituitary gland and hypothalamus that we don't need to make any more LH or FSH. Um, So that's why you'll see like LH and FSH levels drop off after ovulation.
LH. Okay.
Yeah, here's another picture here that actually shows like the levels. So you can see like the LH, FSH are generally higher during that follicular phase.
And
Um,
then it drops
that-
off.
yeah, yeah. And then for the estrogen and progesterone, you can see that they kind of like or rather the progesterone goes goes up during the development of the corpus luteum. So that's
Okay.
the corpus luteum like actually making the progesterone and the estrogen rather than it being, given off by the ovaries from the LH and FSH from the anterior pituitary. And the thing that makes the corpus luteum able to produce progesterone is human chorionic gonadotropin. So, and that's actually produced by the placenta.
Interesting. Okay. So to kind of recap there, like the one that really kickstarts the progesterone is LH, but the one that continues in this case is HCG, as you're saying, by the placenta, you're saying.
Yes.
Okay. All right, cool.
Yeah. So all in all, progesterone is used to maintain that uterine lining, the
Yeah.
endometrium. You could kind of see that in like that second picture, but yeah, that's what uh that's what progesterone does. And yeah, so you could see on the, after the during like the luteal phase, you can see that the uterine lining gets more, you know, vascularity and also gets thicker.
Yeah.
And that's to prepare the uterine wall for implantation of the developing embryo if
Mm.
there's fertilization. So that happens now, and that's what progesterone is needed for. Um, and so
Okay.
yeah, and so HCG, so the luteinizing hormone that you no longer, get after ovulation, like the luteinizing hormone, um
Let me see.
uh
There's a blue. But
yeah,
yes, seems like as the second of like the follicles release, yeah,
yeah,
stuff goes down
yeah. So like
drastically.
HCG kind of mimics the
Mm.
action of luteinizing hormone um in binding like it can bind to the luteinizing hormone receptors on the corpus luteum.
Oh, interesting.
Yeah, because like LH binds to the ovary, and in a theca cell, it causes cholesterol to convert into testosterone, which then converts into estrogen in the follicle cell, all within the ovaries. And that production of estrogen can cause negative feedback, but also the corpus luteum can also make the estrogen and progesterone, uh, you know, while it's developing. Um,
Okay.
yeah. So when I say while it's developing, I mean like after the ovaries have already stopped doing it.
Okay.
Yeah.
That makes more sense. All right. Oh, yeah, this one was. Is this a, is yeah, like how should I go about this? I know PDH, all I know is that it turns pyruvate into acetyl-CoA, but the actual aspects to it, I
Mhm.
don't know.
Yeah. So this is an important uh thing to know. And so basically what you have is in the pyruvate dehydrogenase complex, you have three um enzymes. And
Okay.
yeah, so you first have your pyruvate dehydrogenase, which would, so did I ever show you like that generalized redox pathway? The biochem like generalized redox pathway?
No, this is the first time we're touching bio. We've just been doing cars.
Yeah, okay, okay. So do you happen to have the PDFs folder open?
Yeah, I got you.
In there, there's a PDF called uh, Biochem Redox.
Metabolism, brain dump, no, scan.
Biochem Redox. Okay, so we know in we know in Gen Chem redox is like oxidation is loss of electrons, reduction is gain of electrons, right?
Yes.
Now in biochem or like orgo and stuff, that's not very useful because that definition is useful for if we're talking about the oxidation state of like a single element, but not really useful in this regard. So what we're gonna see is instead of using that definition for oxidation, we're gonna use either there's three things that can happen. Either loss of hydrogens, gain of oxygens, or gain of bonds to oxygens. Okay? So first
Lost
start
intelligence
off
in the those. Okay.
yeah. So let's first start off with just this regular alkane over here. So and left to right is oxidation. So when we oxidize this, we're going to remove hydrogens to get this with that double bond
Mm,
to get the alkene. Okay. Then we oxidize that again. And so this time we're going to gain an oxygen. So this would really turn into this, like that, like an alcohol.
okay.
So if it's a tertiary alcohol, you cannot oxidize that any further. If
Yeah.
it's yeah, if it's a secondary alcohol, you can then gain bonds to oxygen to get a ketone. While you're
Mm.
at a ketone, you can't go further. Can't oxidize further. If it's a primary alcohol, you could oxidize it into what's this?
Into what are you pointing at? Yeah, that's aldehyde.
Yep. Now that can also be further oxidized into, so we add another oxygen for this. What's this?
Carboxylic acid.
Yep. And then we add another bond to the other oxygen to get what's this?
Carbon dioxide.
Yes. Good. So now PCC is a weak oxidizing agent. So it's able to oxidize the alcohol into the aldehyde, um, or the secondary to the ketone, but it can't do anything further. But KMNO4 is a strong oxidizing agent, which means it can oxidize it all the way to the carboxylic acid. Now, going
Mm,
from right to left here is reduction, and there's similar things for that too. It actually is more common to see. So, for instance, to go from here to here, you can use sodium borohydride, which is a weak reducing agent.
mm,
But if you want to go from here to here, you'd use lithium aluminum hydride, which is a strong reducing agent, which can, so the sodium borohydride can only reduce ketones and aldehydes. The lithium aluminum hydride can reduce ketones, aldehydes, carboxylic acids, amides, esters, pretty much everything.
I thought it was okay. Yeah, I thought it was just the ketones, aldehydes, and carboxylic acid. I didn't think about the derivatives, but
Yep,
okay,
yep.
interesting.
Yeah, yeah.
Okay.
So if we take a molecule of pyruvate, it would look like this, right?
Yes.
Okay. Now, if what like let's say that we oxidize this, what can happen? Let's say that we oxidize it.
Okay. So, I mean, maybe this side becomes like a carboxylic end. Or wait, no. Oh, this gets released because the CO, this carbon dioxide.
Yeah, because the ketone here can't be oxidized further.
Yeah.
But this carboxylic acid can be oxidized into CO2
Hmm.
to release CO2, right?
Yes.
Okay. Now we can go back to that question because
Wrong one.
mhm,
Okay.
yeah. So okay, we know that pyruvate, so like there's like three kind of subunits with three enzymes and stuff. So like first we know that there's like this type of reaction. Um and so I remember when I was first learning this, I was like, okay, we're losing a carbon dioxide, so it should be a pyruvate decarboxylase. And it's funny because like even when like you like look it up, a lot of things that come up, like if you like Google image search it, a lot of things that come up get that wrong too. Like they say that it's a decarboxylation reaction. And it certainly looks like it because you get a carbon dioxide at the end. But
Mm.
but now we know that you can also get that from oxidation of the pyruvate. So that's going to be your first enzyme. It's going to be your pyruvate dehydrogenase. And dehydrogenases we know are oxidoreductases, right? They participate in
Yeah.
redox. So that's your
Mm.
first enzyme. You're also going to get, so you're going to get two dehydrogenases and one um, acyltransferase, which is going to just be the thing that transfers the um, acyl group um, to the uh, to make the acetyl-CoA. So, okay, so pyruvate
Mm.
dehydrogenase, and then you'll have dihydrolipol dehydrogenase, and that's gonna
Okay.
cause like, FADH, um, NADH and stuff. And
I
you'll
mean,
also have, yeah.
from what I remember, isn't like the that whole complex and making pyruvate into acetyl-CoA, I never recalled it making like something like NADH or FADH like cofactors, right?
Oh,
Because if that's dehydrogenase, wouldn't that mean that there's like some FADH or NADH there?
uh-huh. Yep, yep.
But I've never seen that. Is just an exception to the case where there's none of that cofactors being made, or
Oh, well, that stuff does get made, in the pyruvate dehydrogenase complex. Like you do
Oh,
have like NAD plus, turn into NADH, right?
interesting. Okay. Yeah, I didn't know that. Okay.
Yep.
Gotcha.
Because, you need, if we're oxidizing uh the pyruvate, we need something that gets reduced, right?
Yeah, makes sense.
Hmm.
Okay.
Yep. So that's
So
gonna give you, yeah. So yeah. So then you have the dihydrolipol dehydrogenase, and then you're gonna get your transacetylase over there. And yeah, that's pretty much that. So I would just remember like that. So first of all, I would remember that there's two dehydrogenases, and that, you know, the first one is the reason that you get a CO2 come out. And then you just have a transacetylase.
I still don't get the trans. It just oh it converts it gives the remaining pyruvate molecule the CoA.
Yeah,
Okay.
it's the dihyd it looks like um it has like the two SHs, like the two sulfhydryl groups.
SH. Yes. I think I know what you're talking about.
Hmm.
Yeah. Okay. And then again, what does the last dehydrogenase do? Just the dihydro.
Uh-huh.
What does that one do again?
Yeah, so that can so since um the lip the what the dihydrolipoamide has the has two SH groups. So when this turns into like meaning when we oxidize this, we would get
Sulfur bonding to each other?
just that.
Nice. Okay.
And as this happens, the dihydrolipoyl dehydrogenase will uh have FAD turn into FADH. And when this comes back, it can make from NAD plus to NADH.
Okay. Hmm. All right then.
Yeah, let me see if I could find like a good picture of this. We have, okay, we have in the chat. So, okay, so we have our pyruvate here, and then we have this thiamine pyrophosphate, which we don't really have to particularly worry about in this case, but we have our pyruvate dehydrogenase cause the pyruvate essentially to attach here to get it there, and we get CO2 come out,
Hmm.
right?
Yeah.
And then this, structure gets so this is the lipoamide, and yeah, you do have disulfide bond there, and that's why the dihydrolipoyl transacetylase is going to involve the breaking of a disulfide bond and then an acetyl group getting transferred onto it. So we see that it gets broken here, and then the acetyl group
Oh, okay.
gets added
Interesting.
there. And then you have your, I mean, you're still like it's still doing this, like the dihydrolipoyl transacetylase to give
Okay.
you your then we add CoA, coenzyme A with its SH group, and then we add it to make our acetyl CoA, and this molecule, this part is the acetyl part here. So the CoA binds to that, and so we see that this gets kicked off to get this, right?
Okay. And then the cycle repeats, I guess.
Yeah, and then that's where the dihydrolipoyl dehydrogenase comes in because it basically oxidizes this to recreate this. And as it oxidizes it, FAD gets reduced to FADH2, and
Hmm.
FADH2 can also, when it oxidizes back into FADH, cause NAD plus to get reduced into NADH.
Okay. Let's screenshot this.
Yeah. Let me erase my thing.
Oh, I took it right
Okay,
into
okay.
I wanted to see
That,
your thing,
that's fine.
so
That's fine.
okay,
Yeah.
awesome.
You're gonna have to plug your computer in.
Oh shit, you're right. Okay, give me a second.
Yep.
Thanks. Oh no, it's just definition. Hmm. Okay, I think we can go over this ketosis.
Okay. I mean, yeah, so ketosis, like the ketogenic diet and stuff, like ketosis is when you you're not supposed to eat carbs. So because normally carbs, break down into glucose, which our body uses, and our brain can only use glucose and ketone bodies. So, and ketone bodies come from breaking down like lipids. Um, so A says when a body is not supplied with enough lipids to burn, it's like lipids are what you will burn if you're under ketosis. So yeah, so they wrote it's when you don't have enough carbohydrates to burn. So that's
Okay, I see the distinction. Okay.
yep. And then it says B, ketosis and ketoacidosis are the same metabolic states. Ketosis is when you make ketone bodies for an energy source. Ketoacidosis happens when you have an excess of um, acid produced, which could happen if you have a too much like it can happen to people who are diabetic, and that's just when the ketosis process. happens like happens too much and too fast. And therefore, when they say a pathological state means that it's abnormal and it's bad. Because yeah, ketone bodies can an excess of the ketone bodies will be acidic and cause um your blood to become more acidic. So that's what ketoacidosis is. It's just ketosis taken to the extreme, producing a acidic blood, basically. Right.
Okay.
And then C, ketone production is greatest during the early parts of a fast. The early parts of a fast
They're
in
gonna be gluconeogenesis, right?
uh early parts of a fast would be like so you use your glucose in your blood and then you break down glycogen in your muscles,
Yeah.
liver rather, um,
Mhm.
to make glucose. And after a certain amount of time, you're going to start, once you've already depleted your glycogen stores and stuff, that's when you will start burning fat.
Okay, so it's more like later parts of a fast, if anything.
Yep.
Okay.
I have a perfect thing for this. Did I share with you this um, cheat sheet from Jack Weston?
No.
Okay. Yeah, so it's a really, really good one. And I'll tell you what page to go to. Let me see if I can find it.
Oh, damn, so many. Right, biochem, let me see. Ketogenesis, ketolysis.
Yeah, there's something here that would just show like over time the stuff that gets broken down.
Search, search the result. Is there anything on this page that says ketosis? Nope.
Okay, whatever. I'll find it and then I'll tell you when.
Okay.
But yeah, we can.
Save this though.
Mhm,
Small ketones are soluble in water, but larger ones are not.
mhm.
I have no idea how ketones can get bigger.
Yeah. So, all right, whatever. Okay, let's see. Small ketones are soluble in water. Larger ketones um are not. Yeah, that's a pretty weak answer because there's like the ketones that we make in ketosis, there's like three and they're pretty small. but D is kind of just saying like a general statement about ketones in general. So in general, like the longer a carbon chain gets, the less soluble it will be in water.
Yeah, that makes sense.
Yeah.
Okay. I thought ketones all come in the same size, but I guess not.
So like
Interesting.
the ketones associated with ketosis, are small. Um, but I think what they're saying is just they're just saying like generally any ketone uh would be less soluble the longer the chain is.
Okay, I see what you're saying. All right. Okay. Mistake. Um, yeah, I went over these. Oh yeah, this made a mistake. Um, yeah, never mind.
I finally found what I was looking for. It wasn't in the cheat sheet, it was just from the website, but yeah, open this up. Here we go. So yeah, on the
I see.
bottom, you could see, time x-axis, y-axis glucose used, and you can see that we start off with just glucose, um, you know, as as the black line that just goes down, at a constant rate. And that's just the blood glucose that you have, the glucose that you have, right? Once that's
Okay.
done, that's phase one. Once that's done, you enter phase two, in which you have your glycogen start to break down into glucose, so you can use the glucose. Um, and you also you're starting the gluconeogenesis process too. Like you start it the second we run out of glucose here, and it kind of steadily increases. Um,
Gotcha.
Phase three is when your glycogen has already been essentially used up, and gluconeogenesis reaches a peak. But then
Okay.
after that, it's just prolonged starvation, and that's when the um, things like fat breakdown happens. And that means that, you know, you're going to get your ketone bodies and stuff coming in.
Okay.
Mhm.
I guess we can do this kind of as a review. Uh, reading. I think this kind of goes with how to read STS page stuff.
Yeah. Okay. So we protein X is a hexamer composed of three GP1, GP2 disulfide linked heterodimers. So I'm going to actually draw it like with the disulfide part here. So, okay, let's do it like this. Let's say that the weight of GP1 is I'm just gonna use some easy numbers. Let's just say 5. Let's say the weight of GP2 is 2. Okay.
Yeah.
Now we're gonna have a native gel, an SDS, and then reducing gel.
Okay.
And let's see, let's say that the three of these can be attached to one another with like not a disulfide bond, but just like through like intramolecular forces. So we have another one here.
Okay.
That.
Or because it says is you're saying that because it says a hexamer up here?
Yep.
Okay.
But I'm kind of making up the thing about these intramolecular forces here. But okay, so the native gel is the protein as it is. You're not doing anything to the protein, so it's gonna be that hexamer, which means that, well, what weight would it have?
5, 10, 15, 17, 19, 21.
Yep. So we can have a band here at 21. Okay. Now SDS, aka denaturing, means that we're gonna break the intramolecular bonds. We're gonna basically break up all the bonds that aren't disulfide linked. So
Okay.
so that means we would break this like these.
So you just get 3 bands at 7?
Now that's the thing, right? So it will be at 7. Oh, and like this gel is going down, so smaller things go down.
Okay.
But since these are all at 7, it'll it'll just be one band because it's the same weight.
Okay.
All right.
And it
Now,
would be thicker, right? Usually it would be thicker, right?
yeah, it can be thicker, yeah.
Okay.
But that could also be, well, usually they'll ask like how many bands, less so the thickness part. But yeah, okay. Now
So you're
BME,
saying.
yeah. So BME, beta mercaptoethanol, is a reducing agent. So, and that would break the disulfide bonds, right? It would break these.
Yes.
So what should we see for that?
Um, so we'll probably start getting stuff at what? And a 5 and a band at a 5 and a band at a 2, I guess.
Exactly. And sure, we have three things, at 5 and three things at 2. But
Hmm.
yep, it'll still be just two bands.
Okay. This makes more sense.
Yep. So yeah, they love this is high yield, so because they love to ask questions like this. So
Yeah.
yeah,
Yeah, it always gets confusing with the STS and then once they add the reducing, but I think I think you simplifying it to just STS is just and breaking down anything that is not a this is
denaturing,
a little easier.
right?
Yeah.
So so we can put it this way, right? So a protein folds spontaneously, right?
Yes.
So denaturing means you unfold it, right?
Hmm.
So if I removed the SDS from this, right? Uh it will turn back into this hexamer spontaneously.
Okay.
However, like so the reducing agent here is BME. If you remove the reducing agent, it will not reform these bonds, the disulfide bonds. Why do you think?
Because those bonds are broken, you would need some sort of like some other
Ah,
oxidizing agent to cause that to happen.
there you go. You got it. Awesome. Very good. Very
Okay.
yep.
Yeah, that makes sense. Ah, yes. I remember this one. The yeah, the factorials.
Oh,
Yeah,
how many
something about factorials, the
sequences?
fragments. Yeah, I remember there was a couple of questions on one some of the full ends on like something like this too.
Mhm.
The fragments and stuff. Yeah, I didn't it was very hard for me to understand how to go about these type of questions
Got it.
and when to use factorials.
Yeah, so this is what, you would think of. So like for instance, we have four unique fragments, right? And so I'm just thinking of like four slots, right? And
Yeah.
there's like how many sequences can exist. So like and there's four nucleotide types, right? So we can
Mm-hmm.
have like, okay, four possibilities here, like A, T, G, C. You have all four possibilities here, right?
Mm-hmm.
And then once you use one, you have three possibilities left for this slot. Yeah, and you've used another one, two possibilities, used another one possibility. So that's how you would uh
Okay.
do that. Hmm.
And then we just multiply or sum it up or
Oh yeah. So because you have four possibilities here, three possibilities here, two and one here. So then you would multiply them for the total amount of
okay.
possibilities, right?
Okay. So is there any because yeah, like in the answer choice it talks about solving for the factorial.
Yeah, so
Um
this happens to be 4 factorial,
okay.
but it's
So
hard. Uh-huh.
when do we use factorials versus like let's say like 2n or something or
Oh,
1n or something to like an
so
exponent?
so let's say that we had like something like this where they ask um how many, I guess, configurations can this molecule have? Or how about this? How many, if they ask you how many stereoisomers can you make from this,
Mm-hmm.
right? So you take a look at, so stereoisomers means that we're going to be looking at the chiral carbons. For instance, this one, right, is going like whatever, like into the page, out of the page, whatever. Like
Okay.
I just to make it easy, like I don't do like the wedges, I just make one black and one white. So let's say that this chiral center here, it has two possible configurations. It can be like the OH group can be out of the page or into the page, right?
Mm-hmm.
So that's two combinations there. And here as well, there's two combinations. This NH2 can go into the page or out of the page.
Okay.
So that's where the two comes in. And then if we just had this, we would have just two stereoisomers, right? But we have this. So this N value is the number of chiral centers. And the reason that this is two is because a chiral center can only have two possibilities. It can either go into the page or out of the page.
Oh, okay.
So that's
I
where,
see why this. I see why this. That probably wouldn't apply to a question like this.
uh,
Okay.
yep, yep, yep. So yeah. So, but they, so that's where that comes from. Um, But yeah, when it comes to uh combinations like this, where there's different, like it's not just going to be based on the number two, it'll be based on other things. So for instance, the DNA sequences here, like it could also be kind of confusing because like the four comes from like does it come from like four fragments or does it come from four possible nucleotides?
Mm.
It's both in this case. It's four mostly because of the nucleotides. There's four nucleotides, but the number of slots is the what they mean here by the four unique fragments. So let's change the number. What if we had five unique fragments? That means we would have five slots, but it would still be four possibilities here, three here, two here, one here, and like, well, I guess that's why they didn't make it five, because obviously this can't be anything. So let's say that this was a three. Okay. Because the way that I did it that way, like there wouldn't be any possibility for that. Okay, if we had three, so three slots, and then it would just be like four combinations here, or four possibilities here, three here, two here, and that's it. So that's going to be four times three times two, which isn't a factorial.
Okay, because
Well,
factorial, you're always going all the way down to one.
yeah, yeah, JK. It is, because you can have your that one. If you multiply it by the one, it doesn't change anything, right?
Yeah.
So, but but yeah. So here where the number of fragments, think of that as the number of slots, and that
Okay.
the numbers that you put into the slot are the for this the different nucleotides. Yeah.
Okay. Sounds good. Okay. Next one, quickly go over real-time PCR last bit.
Hmm. Okay. Yeah, I mean, well, it's just
It's
you know,
just breaking it up, denaturing it, adding the primers, adding nucleotides, and then annealing, and then making that, right? That's all it is, right? Real-time PCR?
yeah, it lets you amplify some DNA fragment. So and yeah, polymerase chain reaction. So uh so it's like in let's say like in the 70s or 80s, like I was watching like some serial killer like documentary or whatever, and they were like back in the early 90s even like they would if they have from the scene of the crime like some DNA, a small amount, um, they didn't really have a way to analyze that or sequence that DNA without using up that material. So they like save it until the technology becomes better. And once we discovered or made PCR, we were able to amplify that a DNA fragment, meaning that we don't need to have that much to begin with. And also, when I used to work at an HIV/AIDS clinic, and the thing that we would have our patients get tested for like every week almost would be like T cell count, and also viral load. And viral load is basically looks for the virus like in your bloodstream and stuff. And we did
Okay.
that using a PCR, so it can detect like a very, very tiny amount of that virus. So, okay, so now using it for all purposes except, so it has to do with um DNA. I mean, there's could be an RNA part too, because you have the reverse transcriptase thing that you can make. Uh, you can make RNA turn into cDNA and stuff. So
Hmm.
Yeah, like all of these are, and this is also just a good general strategy. Like, what's the most different? Like, this is involving nucleotides, which means DNA and stuff, genes. This is nucleotides and genes. This is nucleotides and genes. This is the only thing that isn't,
Okay.
right? Because while you might have the gene to be translated into a protein, that gene can be expressed or not expressed, right? So, in addition to, so whether it's expressed or not expressed means that we don't know if it's going to be any amount of the protein or some amount. But in addition to that, we don't know like how much of it we would have. So it would be wrong for both of those reasons.
Okay. And how can it identify a mutation at that point? Like based on the fact that it's not expressed or something like a random nucleotide sequence that isn't the normal sequence, I'm guessing.
You're saying why wouldn't it
Like
be?
how is it able to identify the presence of a mutation?
Oh yeah, well, because you're basically using, DNA to, so like let's say that there's a mutation that you want to like look into, you can use PCR to maybe create like complementary DNA that can be uh associated with the DNA that you're like, like unraveling, and then you can have it look for that. It's the same thing that you do for, uh like if you can detect a specific nucleotide sequence, right? You can certainly identify if there's a mutation there.
Yeah, that makes sense.
So basically, like think about it in terms of and this is true for a lot of things, like on the MCAT, like are we talking about like DNA, RNA, like the different biomolecules? Like, are we talking about proteins? Are we talking about genes? Are we talking about RNA? Are we talking about, that kind of thing? So,
Okay,
hmm.
cool. I think there was one more thing. Let's see. Was it this? Yeah, just I didn't realize this, but a lot of Jack Wilson has a lot of these single discrete questions, which have been pretty. I didn't know that exists. It's
Yeah, they have really good ones for the math and stuff as well.
for the math. Let me see. Which one are you talking about?
Oh no, I mean like the ones that you have, but like I think they changed the name, but I think if you go to like sections over here and all the way in the bottom for like discretes, you can click maybe CP. Yeah. So physical discrete set 21, discrete set 7. Like they used to be just called math, but I think they just changed the name, but these are pretty good.
Okay. Yeah, I might try doing more of them. Is there a section you think that is not worth my time or no?
A section that's not worth your time, like
Yeah,
in
like something that I probably shouldn't worry about doing or shouldn't really try to do, or do you think all of them are helpful? I mean, do you think there's anything that I shouldn't like really bother doing or something?
oh, I mean, like so it depends on what you like. So like for the content, so like the question you're just about to ask me, right?
Hmm.
We can look at that, but yeah, it would be based on like, what you know. So like the content diagnostic shows the things that you know that you don't need to worry about, and the things that don't know, right? It'll just be based on that.
Okay.
So,
Yeah, honestly, my only real question is like I understand most of this,
mhm.
but last thing is like I was trying to understand the aspect of a dielectric
Mm,
and a capacitor. And from my understanding so far, I want to tell you what I understand. It seems that it opposes the electric field from the overall capacitor, and that apparently increases charge. But
okay.
I can't understand the relationship between the electric field, increasing charge, and then dielectric. That I don't know why, but that stuff keeps confusing me.
Got it, got it. Yep. So a dielectric is going to oppose the electric field of the capacitor, which makes that overall electric field strength to be smaller. And what that can do is, well, um, it can prevent that discharge from happening like immediately and allow more of a charge to build up on that capacitor's surface. So so here they're showing us parallel plate with a dielectric. So we first have one without it, and we have this like electric field charge per unit area over permittivity of free space. Um, and when we add the dielectric, which is, something that's going to be polar, it'll
Hmm.
align such that the negative ends will be aligned near like the positive plate, and the positive ends of the dielectric will be aligned with the negative plate. And that just makes the um, electric field part smaller. And since we have a constant voltage, the only thing that can change then is the charges that build up on the surface of that capacitor.
Yeah, that last part is a little confusing. I mean, I guess because you're saying essentially these charges oppose, like this plus aligns with, it's like these negative charges align with this positive end, and these positive charges align with this negative end. And you're saying that balances out the electric field?
Yeah, it basically makes it reduces that the electric field strength.
Okay. But if that's the case, then how is it able to hold more charge?
Oh, because it makes it so that it's not going to be discharged as easily. You are trying to see if I can.
So it's harder for the charge to move between the plates?
Mhm.
Interesting. Okay.
Yeah. So the um it acts as like an insulator.
Hmm. Okay.
So that can prevent the conduction between the plates. And that means that you can basically So I'll write it like this. So basically what happens is that the dielectric reduces the electric field between the plates and
Okay.
that makes it because if we had something like this, we would have like, electrons going past because both the plates are conductors and we just have free space between them. So, charges will build up on a plate and then get discharged through the free space. But if we put an insulator in between the two conductors, it makes it less likely for it to, discharge. So you get to charge it up some more.
Mm,
Yep,
okay.
charge it up some more, and that means that you can have more electrons, you know, on that plate. And
Hmm.
since capacitance is like essentially like number of electrons over like per volt, um, increase in that number of electrons that gets charged will increase the capacitance. And if you increase the capacitance, you could also increase the energy stored in the capacitor because of that equation. Like potential energy of a capacitor is 1/2 CV squared.
Right.
So you'll have more energy that can be stored as well. Um, and yeah, you can even make it like because also like capacitance would be like proportional to area, like area of the plate. More area means you can put more, char store more charge on it.
Yeah.
But if you add a dielectric, you can you don't need the capacitor to be as big because you could increase capacitance through this other way.
Mm,
Hmm?
I see. Wow, okay.
Yep.
Cool. I mean, yeah, that was it. That was it. That was it. Let me get my phone. I'm just gonna be today.
What's up?
How much is it gonna be today?
Oh yeah, 113.
Okay. Yeah, I just sent it.
Okay.
Can we meet next
Yeah, one.
Wednesday?
Um, I'm free after 6 if that works.
Right after 6.
Yeah. Wait, you said Wednesday, right?
Yeah.
Yeah. Wednesday, I have basically people from, you can't do like after 6 or anything.
Yeah, we can do it. What time after 6?
Oh, like anytime. Because I have students from like noon through 6. So maybe
Okay.
like 6:30 or something.
6 o'clock on the dot, and then we'll just go from there.
Well, so I end with a student at 6,
Okay,
so
6:30 then?
maybe like 6:20 or something.
Yeah, that's fine. Yeah.
So, and I would I guess like after this session, like you have a Calendly link, right?
Okay, yeah.
You can use that to schedule for future sessions. That way you can get the slots before other people take them.
Okay. See what you're
Right.
saying. Okay.
So here, so we said 6:20,
Mm-hmm.
and how long did you want that to be?
Yeah, I think we can make it 2 hours.
Okay. Okay. So Wednesday the 27th at 6:20 Eastern, and what will you be doing between now and then? Was it like a full length, or you're gonna wait for this section B?
Yeah, I'm probably gonna have probably my plan is just to finish the entire section by then.
All right, awesome.
Just get back on cars again. Do a lot of cars.
All
So
right. Yeah,
that's really
awesome. All right. Have a good weekend. I'll see you on.
good. Take care, man.